Hello everyone, this is our last lecture of biological classification. In the earlier sessions, what we studied was kingdoms. We studied kingdom, Monera, we studied Protista, and fungi. The agenda of our today's session will be the recap at first, then we'll have a view on a bit of kingdom plant and kingdom animal, which we'll study further in our next classes in next chapter, and then we'll study about viruses, viroids, and prions. So this is what we'll study in our today's lecture. At last, we'll study about lichen. Let's begin. So in earlier sessions, we studied about R.H. Whitaker gave the 5-kingdom classification, which was 2-domain system. Further, we studied the 6th-kingdom or 3-domain system. Then we studied about kingdom Monera, and in kingdom Monera we classified organisms. We classified the bacteria as it is the sole kingdom, in which we classified bacteria into archaebacteria having a glycoproteinaceous cell wall, and eubacteria having a rigid cell wall. Eubacteria were further of two types on the basis of stain gram-positive or gram-negative. We studied about cyanobacteria, we studied about cyanobac, then we studied about archaebacteria, and archaebacteria are basic bacteria, which includes methanogens found in marsh gases, marshlands, marsh gases methane, and in gut of ruminant animals, we have halophiles salt-loving, we have thermocerophiles, which are found in hot sulfur spring pools. Cyanobacteria have chlorophyll A, they are photosynthetic autotrophs. We studied about nostoc, anavanna, and oscillatoria, which are responsible for nitrogen fixing. They have specialized cells for nitrogen fixing known as heterocystic cells, and cyanobacteria's outer layer is a mucilaginous sheath, and that's all about kingdom Monera. Further up, we studied about kingdom Protista. We studied different phylums. At first, we studied chrysophytes. We studied dinoflage, lates. Chrysophytes were diatoms and desmides. If I talk about desmides, they are golden algae. If I talk about diatoms, they have soap-like overlapping, too thin overlapping soap-like, which are embedded with silica. So inside it is silica. So this is regarded as an indestructible thing. Therefore, it is called diatomaceous earth, used for polishing, grating, filtration of syrup and oils. Then we study dinoflagellates, which are of different colors. They are majorly responsible for causing red tides. They have marine habitat. Gonulax is the example, and they release toxins which kill marine animals and fishes, and their outer surface is cellulosic. Then in kingdom Protista we studied about euglenoids. Example is euglena, majorly photosynthetic and heterotrophic in absence of light. Lacks cell wall. Euglenoids lack cell wall. However, a proteinaceous flexible layer called pellicle. Called pellicle is present. They have same pigment as that of the higher plants. Then we studied about slime molds, saprophytic on dead twigs and leaves, saprophytic on dead twigs and leaves, and under favorable conditions, under fav conditions, they form slime molds, form aggregation called they form aggregation called plasmodium. Under unfav conditions, they form spores having true walls, can survive for many years, extremely resistant, and yeah, they are dispersed in air currents. Then we studied about protozoans, amoeboid. Pseudopodia was the characteristic false feet. Pseudopodia, false feet, they spread their false feet and engulf the food. Number 2 was they have silica shells. Examples are amoeba and entamoeba histolytica and E. histolytica. Then we have flagellated. We have trypanosoma, known for causing sleeping sickness. Parasitic. And then we have sporozoan, which are parasitic. Example is plasmodium. They have infective spore stage. Then we have ciliated protozoan, whose example is Paramecium, and the major characteristic is they have gullet. Their water is stirred when they engulf the food. So that's it about protozoan. Now we are moving ahead with the kingdom fungi. Kingdom fungi have phycomycetes, ascomycetes, have basidiomycetes, and it also has deuteromycetes. If I talk about phycomycetes are the most basic one. Okay. Now we are going to talk about the habitat. They are mostly found in aquatic places, damp places, log of wood, sherry places. They are obligate parasites. They are saprophytic. Ascomycetes are saprophytic, corpophilous, and parasitic. They are saprophytic, they are parasitic as well. There is not much mention about the deuteromycetes, about their habitat. However, sexual reproduction is absent in them. So when the true sexual phase is found, then they are classified into asco or basidiomycetes. Further on, we are going to study about phycomycetes, that they have choanocytic and aseptate hyphae. Choanocytic means long continuous tube filled with multinucleated cytoplasm. Ascomycetes have septate and branched mycelium. Septate and branched mycelium, basidiomycete, and again deuteroseptate and branched. Now I'm going to talk about the mode of reproductions. If I talk about the mode of reproduction, the phycomycetes have a sexual mode of reproduction, and vegetative by fragmentation. So let's begin. If I talk about the asexual mode of reproduction. It is carried out by either motile spores or non-motile. If I talk about motile, the example is zoospore. Zoospore for asexual and motile. If I talk about non-motile spores, we have aplanospores. We have aplanospores. We have aplanospores. If I talk about sexual reproduction, if I talk about sexual reproduction, in sexual reproduction, we have we have zygospores. We have zygospores. So if I talk about asexual spores, these are produced endogenously in phycomycetes. These are produced endogenously in phycomycetes. And then we have examples: Rhizopus, Mucor, and Albugo. If we talk about ascomycetes, they have asexual reproduction by conidiophores. Sexual reproduction by ascospores in fruiting bodies called ascocarp. Fruit body called ascocarp. That's it. And examples are aspergillus. Then we have claviceps, we have neurospora, we have penicillin, we have yeast. The only single-celled organism present in kingdom fungi is yeast. Then we study about basidiospores. So asexual spores are not formed, but vegetative reproduction by fragmentation, and sexual spores are formed by plasmogamy, where two different strain gametes are involved. And deuteromycetes are imperfect fungi. They puffballs and toadstools are basidiomycetes. Morels and truffles are ascomycetes. Deuteromycete are imperfect fungi, and when the true sexual stage is found in them, then they are classified into basidiomycetes. Their examples are Alternaria, Coelotrichum, and Trichoderma polysporum, which is used for production of cyclosporin A. At last, in the kingdom fungi, the last thing that was left was the Asco and Basidiomycetes have a dikaryon stage. Phase is known as dikaryophase. This phase is called dikaryophase and stage is dikaryon. And when this n plus n fuses, so this is 2n, and then the fungi has fruiting bodies which undergo reduction division to form spores, and after that it all happens. Okay, so in phycomycetes there is like directly when two gametes fuse, so 2n condition comes, but in asco and basidiomycete we have dikaryon stage or dikaryophase. That's it. Now we're going to move on to our next topic, that is our kingdom Plantae. So if I talk about kingdom Plantae, they have a unique feature that is known as alternation of generation. Alternation of generation. Alternation of generation. It is basic phenomenon. Alternation of generation is a basic phenomenon in which life cycle of plant have two distinct phases, one the diploid sporophytic and the other haploid gametophytic, that alternate with each other, and the lengths may vary. Okay, so plants have two stages. Plants have two stages. One diploid sporophytic and one haploid gametophytic. This interchange is called alternation of generation. AOG, alternation of generation. Now we are going to talk about some insectivorous plants. There are some plants, rarely some plants, which are partially heterotrophic. Partial heterotrophic. Like bladderwort and Venus flytrap. We have a parasite as well which grows on other organisms. Is Cuscuta. Is Cuscuta. They have chloroplasts. Their cell walls are made up of majorly cellulose, and they have different divisions. As I have told you earlier as well, kingdom के नीचे plants में आता है division. Phylum नहीं बोलते, division बोलते हैं. और इस division में आते हैं हमारे algae, then bryophytes, then pteridophytes, then gymnosperms and angiosperms. So this is what we are going to study in the kingdom plant chapter. Next we have kingdom animal. Animal have holozoic or mixotrophic mode of nutrition, that we take nutrition from plants, from other animals by eating other animals as well. So we are heterotrophic eukaryotic organisms which have an entire body, we can move. Yeah, so all animals show movement but not locomotion. So all animals can move, like can move their hand or different things, but displacing from one place to other is not possible for all organisms, as there are sessile organisms including poriferans. These are sessile organisms. Okay. Further up, holozoic nutrition, that is we ingest the food and then we get nutrition, and most of us are capable of locomotion here. And sexual reproduction is by copulation, male and female, followed by embryological development. That we all know that sexual reproduction is by copulation, and after copulation there is embryological development. So that's all the basic description, where we study that we have holozoic sort of nutrition, and holozoic means a nutrition in which we ingest food. We have mixotrophic sort of nutrition. Mixotrophic means that we take our nutrition from different sources, and at last it's all animals show movement, but some of them do not show locomotion. Majority of them show locomotion. Next up, the thing we are going to study now is virus. Term virus was given by D.J. Ivanovsky. Virus means venom. Virus means venom. Virus means venom or poison. Virus means venom or poison. And these are microscopic, non-cellular. They don't have any sort of cell. These are the exceptions of Whittaker system. These are microscopic, non-cellular organisms. We can't differentiate that they are living or non-living, as when they enter the host body they are living, and otherwise they are non-living. So microscopic, non-cellular organisms having inert crystalline structure, and not crystalline structure outside living cell. Outside living cell. So it was DJ Ivanovsky which named them so, and it was 1892. In 1892 it happened. In 1892, and yeah, DJ Ivanovsky recognized certain microbes as they caused tobacco mosaic virus (TMV). TMV is a bacillus rod-shaped virus. Rod-shaped virus. It causes tobacco mosaic disease, and this is caused in tobacco. So the structure of TMV, it can be asked. It is like grapes. So majorly viruses have RNA as genetic material. So do tobacco mosaic virus have. And then we study that M.W. Bejernik. M.W. Bejernik was the scientist who in 1898 demonstrated the extract of infectious virus that infected healthy plants, and he said it contagium vivum fluidum, or infectious living fluid. Contagium, this is an important point. In contagium vivum fluidum. Vivum, viva means live, long live. So contagium means contagious or disease spreading living fluid. There was virus. He demonstrated it, he extracted the thing from the tobacco plant, which infected healthy plants. They also said that viruses are They also said viruses are obligate parasites. Viruses are obligate parasites as they are parasites only, and they can be crystallized, and crystals are majorly of proteins. So viruses that infect plants, viruses infecting plant are majorly single-stranded RNA, and viruses infecting animals, viruses infecting animals are majorly double-stranded RNA or single-stranded RNA. So they are either single-stranded RNA or double-stranded RNA or double-stranded DNA. So viruses can be of any genetic material, double-stranded DNA, double-stranded RNA, single-stranded RNA in animals. And if I talk about in plants, they are single-stranded RNA. Now we're going to study about viruses which affect we're going to study what viruses, which affect bacteria, are called bacteriophage. Bacteriophage. 74. Bacteriophage. 86. Nucleotides. And lambda bacteriophage. 48,502 base pairs. Now we are going to draw the structure of bacteriophage. I'm not good at drawing. So that's a basic bacteriophage. These are called pronges or tail fibers. These are called pronges or tail fibers. So that's all about bacteriophages. And we just have to know about who discovered which thing. So viruses were actually discovered by M.W. Bejernik, that as they caused tobacco mosaic virus. Then we studied about the crystallization. We studied about contagium vivum fluidum, which was demonstrated by our next scientist, which was M.W. Bejernik, which demonstrated that this fluid is causing the uh, disease in healthy plants. Then we have other scientists, W.M. Stanley. W.M. Stanley demonstrated that these viruses can be crystallized, and crystals are majorly of proteins, and the viruses affecting plants are majorly single-stranded RNA. However, in animals there are single-stranded RNA, double-stranded DNA, double-stranded RNA. These are the genetic materials of viruses that can have. Then we studied about the bacteria that is affecting the virus that is affecting bacteria, that is bacteriophage, and bacteriophages have double-stranded DNA virus. Bacteriophages have double-stranded DNA. Double-stranded DNA. Okay, so if I talk about plants, they have RNA. Plants have RNA. If I talk about animals, sRNA, dsRNA, dsDNA. If I talk about bacteriophages, the viruses which affect bacteria have double-stranded DNA. And then we are going to talk about the small subunits called caspid form capsomere. That is protein coat. If we talk about the tobacco mosaic disease, leaf curling, stunted growth, vein clearing, yellowing and vein clearing. That's it. Leaf rolling, stunted growth, that is the TMV symptoms. Yellowing and clearing of vein, yellowing and clearing of vein. Now in 1971, T.O. Dana. So in 1971, T.O. Dana was a scientist which discovered viroids. So viroids are nothing but these are smaller than viruses, and they lack a protein coat, and they are of low molecular weight. They lack protein coat, low molecular weight, and viroids are known for causing potato spindle tuber disease. Prions are proteinaceous in nature, known for causing C.R. Jacob disease or mad cow disease, and BSC or mad cow disease. In humans, CGD. And I have talked about Lycanaria as well, that they are pollution indicators. Phycobiont and mycobiont. Phycobiont food, that is algae, and myobiont is our fungi, which provides shelter. So that's it, and the chapter ends. Biological classification. At last मैं जो summarize करने जा रहा हूँ. So let's begin. Summary. Diseases caused by viruses. सबसे पहले तो DJ वेनोस्कर ने इसको सर्च किया, ठीक है. वायरस की बात करूँ तो वायरस टोबैको मोज़ेक वायरस, ठीक है. ये होता है आपका सिंगल स्ट्रैंडेड RNA, ठीक है. उसके बाद मैं बात करता हूँ वायरोइड्स की. वायरोइड्स क्या होते हैं? वायरस मतलब they lack a protein coat of low molecular weight, and low molecular weight के होते हैं, lack a protein coat, and जब भी फाइंड किया गया था फ्री RNA, बोलते हैं इसको फ्री RNA. It was found to be a free RNA, and 1971 में T.O. Dana ने इसको डिस्कवर किया था, और ये करता पोटैटो स्पिंडल ट्यूबर. अब नेक्स्ट बात है प्रायोन्स करते हैं BSC या मैड काउ डिजीज, जो कि एनालोगस है, यानी कि कोई ओरिजिन तो सेम नहीं है, बट किस बीमारी की तरह सेम है ये, जैसे कि हमारे में C.R. Jacobs डिजीज होता है, ठीक है. दैट्स इज अबाउट प्रायोन्स. एंड नेक्स्ट चीज़ वायरसेस की आपको ये याद रखने पड़ेंगे कि कब कब किसने क्या क्या किया था. तो अब जैसे वायरस था, इसको डिस्कवर एंड नेम किसने दिया? DJ इवानोस्की ने 1892, ठीक है. अब जो है वो इसको एक्सट्रैक्ट किसने किया? MW बेजर्निक. कंटेजियम वेवल फ्लूइड इसने दिया 1898. फिर WM स्टेनले 1935. WM स्टेनले ने 1935 में क्या किया, ठीक है. तो उन्होंने क्या किया कि तुम्हारा वायरसेस को क्रिस्टलाइज किया, और वायरसेस को क्रिस्टलाइज करके बताया दे आर प्रोटीनेशियस इन नेचर. तो ये चीज़ आपको पता होनी चाहिए. 1971 T.O. Dana. वायरोइड्स RNA होता है, फ्री RNA होता है इनके पास, ठीक है. T.O. Dana ने इसको पोटैटो स्पिंडल ट्यूबर डिजीज करता है. प्रायोन्स जो है वो मैड काउ या फिर बोवाइन स्पॉन्जिफॉर्म एन्सेफेलोथैलेपी करते हैं, जो कि मैड काउ डिजीज के नाम से बोला जाता है, और एनालोगस है ये ह्यूमन में होने वाले C.R. Jacob डिजीज के, ठीक है. और इसी के साथ हमारा जो चैप्टर है वो होता है ख़त्म, और नेक्स्ट सेशन में हम जो है वो इसके क्वेश्चंस करेंगे, MCQs करेंगे, एंड एट लास्ट फिर हमारा बचता है प्लांट किंगडम एंड एनिमल किंगडम, और इसी के साथ ही यूनिट हमारी होगी ख़त्म. थैंक यू. Have a great day.